And I also start to record the webinar.
Okay. Should we start or?
Yeah, we can wait for another couple of minutes until
Okay.
everyone joins, but maybe until this is happening, I'd like to welcome everyone to today's Friday seminar, to the SEM, which is financed by the LABEX CEMEB, and we're very grateful to Guinea for providing us with a Zoom webinar and helping us with the technical side of things. So thank you very much. For everyone who is freshly joining, you can, of course, as with every seminar, ask your questions. So at the bottom of your window, of the Zoom window, you should see a Q&A tab where already during the seminar you can ask your questions, and you can also vote up questions that have been asked by other people so that we have an idea of which questions you all think are important. And finally, there's the chat via which you can contact us or everyone to if you have technical issues or something like that. Yes, I think Finn, if you want, you can start.
Yeah, so we're very happy to host today Simon Segar, who came over here to Montpellier for the pleasure of good weather. Unfortunately, it's not here today, and we're very British. So Simon Segar, he's born in 1982, so he's a young guy of 37 years old, not yet 40. And he started his career in research with a PhD at the University of Reading. Obviously, this was not quite his optimal weather. So well, the study with James Cook was in Eastern Australia on parasitic fig wasps. Then after that, he moved to Europe, so to the Czech Republic, to South Bohemia, where he stayed for 8 years. That was between 2012 and 2019. So, well, South Bohemia may not be the optimal climate for Simon Segar, so his fieldwork was in New Guinea with all the projects that are developed by Wojciech Nowotny. So this involves a lot about well, what he's going to talk about today. So maybe it's not so important that I present all this. And then beyond that, there was something. So this is all about nasty insects feeding on plants, and nasty insects feeding on insects feeding on plants, and nasty insects feeding on insects that feed on insects that feed on plants. So it's trophic networks and things like that. And in between, he had a spell of looking at nice animals, which were insects feeding on plants, but being mutualists. So that was fig wasp. So basically, this is his career. And then last year, he got a position as a lecturer in entomology at the Harper Adams University in the UK. So he's planning to do field work somewhere in Africa, I think, just because Reading is, no, Adams Harper University is not that good climate. So, well, I let him present now.
Okay, thank you, Finn. Okay. So if I share, I'll start to share screen. Okay, so thank you very much for the introduction, Finn. I'm just going to start my timer so that I can not keep to time. I'd just like to say thanks everyone for turning up, and now I'm going to give a talk about some of the work, as Finn's been saying, that we've conducted in New Guinea. So I've been really lucky that I've worked some very fantastic places over the past few years, and been lucky to be involved in some really nice research with lots of good people. So I'm gonna talk about the plan of the presentation. So initially, I want to talk about some of those people and some of the places, the people behind the research that really make things happen. Then I'm going to talk about 3 published papers, so I'll try to go through those relatively briefly, just pulling out the important parts, or what I think are the important parts anyway, and then I'm going to present some new results in section 5. I have a very, very brief section on what, as Finn alluded to, what I'd like to do next, and this is kind of a conceptual extension of the stuff in the talk, but it's on a different system, in a slightly different setting. So first, I'm going to introduce some of the people and the places. So I've been really, really lucky to work in New Guinea. So it's a fantastic place. It's a fantastically diverse place. Sorry, Professor Simon Leather's just ringing me on the other line, so I'm just going to hang up on him. So it's incredibly diverse. It's estimated that about 0.5% of the world's species live. So it's 0.5% of the world's landmass, but about 5% of the world's species live in New Guinea. So New Guinea's got a really high rate of endemism. So like many montane, rugged, tropical countries, it's got a high level of endemism and a high level of species turnover with elevation, which both contribute to these red spots on this map. Okay, so if I get back into this, so Finn also mentioned the work of Professor Wojtek Nowotny. So Wojtek has been working in New Guinea now for 20-30 years, and he's done some fantastic research on all sorts of elements of insect-plant interaction. So I'm going to briefly introduce some of those previous papers as to set up the story for some of the work that we've been doing. But the New Guinea Benetang Research Centre is based in this lovely tropical lagoon. You can see the green circle just hovering over the top in Madang, which is in the New Guinea part of Papua New Guinea, so the north, slightly wetter part of the country. Still quite a lot of dense rainforest, and some fantastic knowledge in the people working there. So often they have a kind of encyclopedic knowledge of the local forest, what the species are, how the interactions are playing out. So we work very closely alongside New Guinean nationals to explore their forests alongside them. And one of Wojciech's approaches is very natural history based. So we do a lot of collecting and rearing of insects on plants. We do lots of morphotyping, lots of DNA barcoding to establish differences between males, females, different species, cryptic species. And a lot of this work is done at BRC and the kind of extension lab. So this sign in the bottom right-hand corner is from Ohu Bush Lab, which is a lab in a nice village called Ohu that where George Weibland did a lot of his kind of fig wasp research. We're based largely there with Bruce Asua. So Bruce Asua is hiding behind that, doing his best to hide behind that big log in the top left-hand corner. He's a fantastic researcher. He's probably one of the best people to work with in terms of figs in New Guinea. And New Guinea is like a kind of Disneyland for figs. They're just everywhere. There's 150 species of fig in New Guinea. Many of them found in the areas that we work in, but Bruce has a fair oversight of many of those species. He's worked very closely with George, so that's George Weibland holding a specimen of Ficus damaropsis in his hand, and of course Wojciech Nowotny. So this is a sort of first generation of fig researchers, and a nice story, a nice part of the story is that if you look in the bottom left-hand corner, the guy, I can't hover, he's got green welly boots and kind of beige shorts. He's standing up with a blue t-shirt and a blue bag. That's Emil Bruce, or Mentap Sissol as he's sometimes known, who is Bruce's son, standing with my PhD student Daniel in a village in New Guinea highlands. So a lot of this knowledge is transferred to the next generation, including New Guinean PhD students like Leggy, who's doing his best to look like he's concentrating on his computer when he knows he's being photographed. So these are some of the people. Some of my PhD students that I've been directly involved in, Dr. Martin Wolf at the top, who did a lot of this work on fig herbivores, and Dr. Daniel Soto Valaris at the bottom, who's now at Prague. He's done a lot of really nice work on the population genetics of some of these species. So I think it's nice to be able to talk about some of the people behind the work and introduce them, as well as just the research itself. So the second part of the research of the talk, or the first kind of part of the research element, is about predicting interactions in lowland forests in New Guinea. New Guinea forest is fantastic. There's no other word for it really. It's incredibly species rich. There's land where it's often in good condition because of the way that the land is owned by a kind of customary land rights. It's quite hard to buy up big logging concessions in New Guinea. So a lot of the forest is in pretty good condition, and you can see it for miles around. And this is a view from Bruce Asua's garden, actually, at the top of the hill, overlooking the butterfly reserve in Ohu. And this is slightly disturbed forest. You can see the palm trees. You can see some gaps developing, but this is kind of typical of this particular region, this particular part of Ohu, of Madang. It's a kind of matrix of primary and secondary rainforest, which is also interesting because it's the kind of agricultural approach is to sort of Swidden agriculture, the slash and burn, where people will kind of fell a hectare of forest, have a garden, and then let that forest regenerate. So you do get a genuine, in combination with a very active tectonic landscape, you get a lot of gaps created in the forest. So you do get a lot of these secondary species cropping up. So it makes it quite an interesting habitat to work in. It's incredibly complicated. So this is a slide that I've stolen directly from one of Wojciech Nowotny's presentations. And this is a food web from their 2010 paper in Journal of Animal Ecology. And it's kind of mind-blowing, really. So this is just a snippet of the whole thing. You can see along the bottom, the red section is the bit that we're focusing in on. This is an interaction web between depending on the data set, upwards of 150 plus plants and 9,600 insects, 200 species of tree. And there are 50,000 trophic interactions in total. So it's a really complicated web. There's been lots of work done on it, but we're trying to really go beyond this descriptive aspect and understand and predict some of this complexity now. So if we break down some of this complexity, we can see that a lot of these plant genera that are present in lowland rainforest in New Guinea, but this also holds true for tropical forest more largely, is represented by what E.J.H. Corner, and as Finn knows, you can't study figs without studying Corner, called his large tropical genera. So that's E.J.H. Corner there with his trained monkey. So his monkey used to climb up the tree and collect voucher specimens for him. I think that's a much nicer approach than the kind of Russell Wallace approach of shooting down butterflies with shotguns and that kind of thing. So it's he's an interesting guy. Alwyn Gentry also kind of came to the same conclusion with his species swarms. And what the message is that a lot of lowland tropical rainforest is represented by these diverse genera. So there are thousands of species of Syzygium and Psychotria, hundreds of species of Ficus, hundreds of species of Macaranga, and a lot of the interactions in these webs focus around these big genera. So if we can understand what's going on in these genera, then we can start to unravel the complexity of these networks. So typically these campaigns are large. So these are some of the guys in the field. I think it's nice to show some pictures of how this all works. So that's a typical field lab. You can see the processing there. You've got people morphotyping insects, rearing caterpillars. There's some photography going on in the corner. And this is a sort of organized biodiversity campaign on an epic scale, really. It's work that I think technically in some aspects could probably be replaced with DNA, metabarcoding, but you don't get the same resolution and the same insights, the same natural history insights as we're starting to get from some of this. And I hope I can pick up on those in some of the last slides. So that's an action shot, really. And if we zoom in even further, so we've kind of reduced our plant diversity. If we kind of get rid of some of the insects as well and reduce our insect diversity, we're going to focus primarily on the larval leaf chewers, and that's a guild that's basically caterpillars. They're interesting because they've got an intermediate host specificity. They're not dramatically host specific in the way that a gauly insect or a leaf sucker is, or a kind of mining insect, but they're not as generalist as a big adult phasmid or something like that. So they're quite interesting because they've got a certain degree of structure in their specificity. And if we scale back up and we look across space as well as and one location, we can see that some of these networks are pretty stable. Might be something to do with the fact that New Guinea is quite young, but basically these networks can be quite continuous over broad patches of tropical rainforest in northern New Guinea. So this is the turnover in community structure across some of the big genera that we're talking about. So what we want to do, what our aim as well, was to try to understand, and this is a spoiler slide really for people who can't follow or don't want to follow the whole thing. We're interested in trying to measure some of the traits associated with these plants and match that with insect community composition. So I'm going to show a lot of multivariate figures, CCAs, PCAs, that kind of thing, quite often imposing the community of insects and then correlating that with plant traits. And in this case, we've got the effect of polyphenols, so things like tannins and flavonoids and that kind of thing on the community structure of insects across 3 of these big genera. And we can immediately see that there's a huge effect of chemical composition on how these communities look. So Macaranga, a euphorb with incredibly high oxidative activity and polyphenol content, has a very distinct community for example, from ficus, which is relatively low. It still has some interesting polyphenol chemistry, but it's not quite as active as the others. So this is the kind of approach we're trying to take. And I'm going to just whiz through some results from a paper published in 2017 where we tried to tease apart. Our aim was to kind of get our heads around the fact that herbivore specialization appears to function at the genus level. So you get quite high specialization for genera of plants, of herbivores across genera of plants, rather than the species level. So you often get genus specialists. But the phylogenetic distance between these hosts doesn't entirely explain herbivore assemblage. So there's something else going on, and we know from multiple studies and a long history that plant chemistry is an important proximate and evolutionary driver of insect community structure. So our aim was to use host use data of insect and plant phylogenies, as well as in ecological traits, to try and predict some of these herbivore assemblages. So here we're interested in bipartite phylogenies, the phylogenies of the insects as well as the plants. And we took an approach, so this borrowed quite heavily on some of the ideas of Ehrlich and Raven, particularly Joy and Krebsey, from some of their, so I started this postdoc in 2012. So a lot of the ideas that kind of ingrained in my thinking were going around at that time. And I think some of them stood the test of time pretty well, actually. There was a lot of thought about how islands were a good model for representing plant genera. So big islands, big genera, lots of species, lots of habitat, phylogenetic isolation, sort of directly relatable to geographic isolation of islands. So we started to think about how we could predict some of this island topography and how some of these distances could help us understand insect plant communities. We also wanted to include natural enemies. We didn't manage to get around to doing that, unfortunately, in this study, and plant defensive traits. And we wanted to use bipartite phylogenies to try to determine some of these drivers of insect plant community structure. Just looking at the time. Okay, so that probably can. So this is you know, I've got 2 young children, so a lot of my time is spent reading pretty high caliber literature, such as The Very Hungry Caterpillar by Eric Carl, and this is an approximation of our scientific approach. We basically wanted to predict host use across the phylogeny of a load of angiosperm plants in New Guinea and the probability of occurrence of particular guilds of caterpillar, species of caterpillar across these plants. Slightly more impressive in some ways representation. This is the angiosperm. This is the angiosperm working group 3 phylogeny, and these are all the groups, the major groups of plant that we sampled. So we tried to, you can see the black circles and the white numbers. These are we basically try to pinpoint members of the entire the diversity of the angiosperm phylogeny. Obviously not the entire phylogeny, that would be ridiculous, but representatives of of species involved for different parts of the branching pattern as our study sample. So in the end, we chose 88 species of plant, and we measured polyphenol activity and content, and we quantified insect-plant interactions, so the community composition, and we focused on, and we generated phylogenies for both plants and insects, and we focused on two of my favorite caterpillar groups. They're not just my favorite because they turn up a lot in my light trap, they're also my favorite because they're incredibly diverse, and they represent two of the most Noctoideae, obviously a very big group, but Geometridae and Pyranoideae, two huge groups of insects. And this is a good representation. We kind of made these co-phylogeny plots, and we tried to match up the phylogenetic information from the plants and the insects. We used a modeling approach, so Matt Helmus and Charles and Ives's phylogenetic bipartite linear models to basically predict the activity, the polyphenol activity. So on the right hand side of that heat map, you've got the predicted activity, and then on the left hand side you've got the observed abundance. And what we could do is we could predict occurrence of presence or absence of some of these moths pretty well using chemical data. We couldn't do it for the pyranoids, but we could do it for the geometrids. And the correlations between our observed models, so the first figure 3, and our observed sort of predictions, our predicted occurrence versus our observed occurrence were pretty good. And on the right hand side, you can see these are just logistic regression plots with varying degrees of data pumped into them. So you've got just the plain phylogeny on the first plot, then we added some trait, then you got what traits on the lower plot, and then when you add traits and phylogeny, so chemistry and phylogeny, you can predict pretty well some of this occurrence. And we managed to do this in a totally unsampled, chemically kind of novel setting in a felled hectare of forest, as well as our plots in the lowland forest. I'm just wary of time. Yeah, you're absolutely right, Emmanuel. It goes a lot quicker when you're doing it this way. So we can conclude that herbivore host breadth is complicated, but it's predictable, and evolutionary history and plant traits seem to be important. And these predictive models might actually be quite useful when we're talking about things like pests, how we can understand the distribution of pest species. So I didn't show the figures, but we actually predicted the host range of some pest species pretty well, even when it came to invasive plant species in New Guinea. So some of these things might be quite useful in a real way as well. Okay, so this is the only picture of a fig wasp. Of course, it's a Pleistodontes, my favorite genus of fig wasp in the center. Someone likened them to the kind of Ferrari of the insect world. I think that's pretty flattering. They're very cool insects. But the rest of the talk is really about fig herbivores. So we're zooming in from looking at a whole range of plants, a whole panel of plants, to specifically looking at figs. And Ficus is known to be a keystone genus, so it's obviously important for herbivores because it's producing fruits all year round. But it's also it's a big deal for insect herbivores as well. So the aim of this particular section was to quantify the caterpillar community associated with 21 species of fig and determine the role of chemical composition in those communities. So the figure above is a bit of a spoiler as well. That's just a PCA, just a simple PCA of community structure of figs with their 3-letter codes, and you can see that you get a large island of community structure, which is actually broadly similar. There's a bit of variation, but you get insects across those hosts that they can share hosts. And then the dots in red are the things which kind of really stand out as being quite distinct. And we're going to try to explore what drives some of these distinctions in insect community composition. So a really quick flyby of the evolution of plant defenses. So this is a huge field. I can't possibly try to summarize it effectively in one slide, but I'll go for it. So there's been a lot of big genus scale studies of the evolution of plant defenses over time. So they're kind of exemplified by Judith Bukhara, Anurag Agrawal, these kind of Mark Johnson, this kind of collection of work that shows there are really two main drivers of plant defensive chemistry when we talk about evolutionary time. Studies from large species-rich tropical genera have shown that in some cases you have a kind of escalation, what we refer to as an escalation of defenses over time. So earlier diverging species in the phylogeny tend to have weaker defenses, and they've adapted to herbivores over time by increasing their investment in defenses through evolutionary time. Other authors have found that actually you get quite high levels of divergence between closely related species. This might be selection for divergence in terms of avoiding sharing herbivores. So we try to test both of these hypotheses, these scenarios in figs. So figs are fascinating, as Finn will tell you, for lots of reasons, but they're pretty fascinating because they've got an interesting suite of plant chemistry. They've got these pentacyclic terpenes. They've got all sorts of terpenes, but these pentacyclic triterpenes probably quite important in defense. They're rich in polyphenols, not as rich as some groups, but they've still got quite a few interesting polyphenols in there. These phenyldrozoene alkaloids are fascinating, so I'm hoping to talk about those in a bit more detail at the end. And they've also got these cysteine proteases, which basically digest the gut of the caterpillars eating them. Often the leaves are quite hairy, not like a septica, but they've got these trichomes. And of course, they've got the kind of normal physical defenses that plants often have. This is a bit of an intimidating plot, but it's a summary of some of the key results. So what we found when we recreated the phylogeny of figs and we mapped some of our traits on those figs was that there was quite a complex distribution of trait values. So in some cases, and we tested for these kinds of these patterns using phylogenetic models, so looking for escalation of plant defenses or divergence in plant defense. And we found that some traits escalated across the plant phylogeny. So alkaloid content provides a really nice example. You can see it at the base of the phylogeny, you've got lower values, and that kind of steadily increases towards the top. There's a clade, the Psychocarpus, the bottom of the phylogeny, that are really active with alkaloids. But alkaloid diversity really takes a huge leap when you get into this clade, the Psychocarpus group. And some of these traits are divergent. So some of them are actually very different between close relatives, more different than you'd expect by chance. This is an example of an escalating trait. So something like oxidative alkaloid diversity and oxidative activity both increase in their values as you go through evolutionary time, as represented by abahost distance derived from the tree. And you can see that really clearly in row 3 of the first plot. You can see that.
Simon?
Yep.
Sorry, can I interrupt? You can use your pointer if you want also, just to let you know.
Ah, okay, excellent. Yes.
This can help for these types of figures.
Yeah, exactly. Thanks very much. I didn't even think to do that. That's a good idea. So you can see, this clade here, this is Psychocarpus. The diversity of alkaloids suddenly takes a huge leap here. And that's an important point to recognize. And what does this all mean for our plants and our insects? Well, it's a complicated story, but in some, we've got some insects which are broadly fig specialists. So they eat lots of other groups, but Corutidae as a whole have evolved to eat figs and a couple of other Moraceae. In our sample at least, and in New Guinea, and they respond to kind of conserved traits. So carutids are pretty good at tracking, they're pretty good at tracking the conserved traits. Oh, okay. Have I got this the wrong way around? Okay, sorry, that's slightly the wrong way around. But what I wanted, what the message is that the specialists, these specialists like the carutids, tend to be very good at eating figs, and they can adapt to it. But it's the traits that are conserved that tend to have an effect on the generalists. So the generalists don't tend to be able to respond so well. The specialists tend to respond a be influenced a lot more by the kinds of traits that are hard to follow over evolutionary time. Okay, so there's a divergent response in herbivores between the lineages. So specialists and generalists basically respond to different different types of trait, and this disparity in trait difference is kind of notable in particular traits. So things like specific leaf area, this doesn't tend to escalate. It tends to be quite different between close relatives rather than escalating over evolutionary time. Whereas some of these conserved traits, things like alkaloid diversity, these are kind of easier for specialists to adapt to. They can track their hosts over time. They can develop mechanisms to overcome these kinds of mechanisms. So alkaloids will be another good example we're going to discuss later on. Whereas generalist herbivores have a tough time to adapt to these conserved traits because often they're feeding on different species. So when they switch back to a host that's got these specialist defenses, they have a tougher time of dealing with them, basically. Sorry, that doesn't help that some of the text has actually been mixed up at the bottom, but I think I've got my point across. Okay, so the main conclusion is that specialists and generalists are basically responding to different types of plant defense, and that shapes these fig communities. So another kind of study on figs. So we were interested in lowland forests, so this kind of this aspect of lowland forests, and we also wanted to know what would happen if we started to study these species along elevational trends. And we know that in New Guinea, elevation is a big deal. We've got a lot. It's one of the most species-rich places in the world, but it's also shares in common with a lot of other species-rich places the fact that it's got these steep elevational gradients. We know there are a lot of montane endemics, and mountains are thought to be important in driving biodiversity in general. So we studied what Emmanuel Toussaint is very sort of nicely referred to as the towering orogeny of New Guinea. So New Guinea is both an incredibly old in terms of its geology. It's part of the Australasian plate, which is obviously got lots of kind of Gondwana and lineages still living there. It's an old, long isolated part of the world, but it's also very new. So it's the kind of collision zone for the Indo-Asiatic plate. So you've got this big crunch zone in the middle, the mountain range that goes through New Guinea, the central range. And our field site, in Madang, you can basically cross 3 different terrain types before you get to our field site. The Finisterre still kind of growing to this day. So it's interesting. It's an interesting place to work in terms of the generation of biodiversity. We work along the Mount Wilhelm elevational complete altitudinal rainforest transect. So it's one of the only sites, only transects in the world that has complete forest across the whole thing. So we work at 6 sites, and these are at 500-meter elevations, and we collect all sorts of taxa from these sites. But in this case, we were interested in figs, and we wanted to quantify the caterpillar and beetle communities associated with 9 species of fig. That's a typo there, and determine the role of chemical composition and elevation in these fig species. So there's another kind of cartoon figure, and you can see more or less what we wanted to do. We wanted to quantify plant chemistry. We wanted to quantify insect plant food webs at various elevations, and we wanted to try to determine how both elevation itself and chemical diversity influence the structure of these food webs. And this was work that was carried out. So a lot of the food web work was carried out by a guy called Leggy Sam, who we saw earlier. So he did a fantastic job of quantifying these interaction networks, and we used some of his data for this particular paper. What did we find? So we analysed 142 trees from 9 species of fig for what we see as two of the most important groups. So these are polyphenols and alkaloids. So I'll point out later who did this chemical analysis and some of the advances. But we basically characterize a total of 29 alkaloids belonging to 5 alkaloid subgroups and 49 polyphenols. So you can think of these compounds at various levels. You can think of them as individuals, so individual compounds, or you can think of groups of compounds which kind of broadly share chemical similarity. So as much as polyphenols and alkaloids are distinct, within polyphenols and alkaloids, you've got groups which share a common structure, which we think may have a common function in terms of their biotic activities. So this plot is a fairly straightforward RDA result. So it just shows us some of the basic patterns. So shows us that elevation, you've got phycoseptamines. These are a group of alkaloids really only found in figs, named for Phycoseptica. They broadly increase with elevation, but you get some quite contrasting trends. And this is a bit of a note and a note that I think it's quite important to kind of say that these total content measures that are sometimes taken are kind of masking a lot of the chemical diversity that you actually find. Equally, a whole kind of metabolomic approach which doesn't take into consideration the groupings, the type of compounds that these are, also loses a lot of the information. So we're trying to kind of push an approach that where you have to really kind of use a bit of chemical knowledge. You can't just use raw metabolomic data. You have to kind of bin these things into groups, but also measures of total alkaloids or total content don't always give you the true story. So here we can see with elevations, polyphenol diversity broadly correlates with elevations. There are some other groups. Unfortunately for you, Pekka, he's really interested in these hydrolyzable tannins. They're not particularly strong, strongly correlated elevation, but they're also not that present in the samples either. But that's just a summary of where we are with how these different groups respond. Okay, same slide again, slightly different title, bigger as well, which is good. Okay, so in a bit more detail, we also kind of ran some mixed models to try to unpick some of these details. And I'm aware that it's a lot of information, a lot of detail on particular compounds, but this will be important later. And I think it kind of shows the need to study these things in detail rather than from a superficial perspective. So some of these compounds, the non-flavonols, showed fairly mixed patterns. So these are things that have protein precipitation activity often, putatively good for mammalian defense. They showed a bit of a mid-peak hump, but no really consistent pattern. Flavanol glycosides, the kinds of things that are involved in UV protection, showed a really strong positive correlation with elevation when you control for species identity. And you can see a slightly nicer way of representing these data here. And in the paper, you can find the species level results as well. Alkaloids are a funny one. So you can see this maybe isn't the ideal plot to show it, but you can see that there's a lot of variation in even just presence or absence of some of these alkaloids. So the mean lies kind of in the middle of. There's a lot of zeros in the data. So we analyze this as binary data actually in the end, but for purposes of presentation, it's nice to see some of that variation as well. Alkaloids tend to increase in diversity as you go up in elevation. So some of these high elevation groups, especially one particular species, is really rich in alkaloids. These different groups have different patterns, but the overall trend is that there's a positive correlation. So what did we find? So again, I'm going to leave this slide up for anyone who's really interested in the detail of the plant chemistry. Obviously everyone is, so that's not an issue. But what we really found is there were particular groups of compounds that were important in determining the composition of these food webs. So for example, alkaloid diversity and phycoseptamines explain not a huge amount, but some of the variation in insect community structure. And then when we break down these compound groups to individual compounds, we can explain also about 20% of the variation in insect community structure. So actually not as high as we might think, not as high as, for example, when we compare across different plant genera. And if we break down what happens over some of the species, so these are different species of fig in different colors, we can see that alkaloid diversity and phycoseptamines have a kind of slightly different loading there, different direction. But the effects are there, the effects are real, but they're not huge. And different compounds respond in different ways. So different compound groups, different compounds themselves tend to have different relationships with elevations and community structure. So unsurprisingly, the answer is it's quite complicated, but we can tease apart some of this here. And I've been told by authorities that there are other organisms out there in the world apart from plants and insects. And one of these are these kind of really cute looking cuscuses, which happen to be very abundant at mid elevations. And we think they might be quite important in driving some of these patterns. We've got no about the fungi. It's likely that some of these alkaloids are important in fungal defense, and these are kind of both things we'd really like to study further. There's just some nice pictures of the transect. I think this river is absolutely beautiful until you're walking through it for about 4 hours and your feet are soaking wet. But these are just some nice shots of the transect before the last part. So I'm broadly keeping to time. This is the last part of the talk. So the aim of this section. So I told you earlier that basically specialist herbivores tend to respond to divergent traits. So specialist herbivores tend to respond to things which diverge between close relatives because they're not necessarily able to adapt to some of these escalating compounds like alkaloids. Even though I cocked up the labeling of the figures. And one of these groups is a really cool looking group of a noctuid called Asota. And Asota is a moth that's found across lots of species in this section, Psychocarpus. And it's basically it's brightly coloured as an adult and as a larva. And we wanted to understand the chemical differences between the caterpillars, the adults, and the frass of this moth to try and work out what it was doing in terms of its chemical ecology. And this work was possible because of all of this natural history and taxonomy work that kind of underpins a lot of our research in BRC. They're really cool looking moths. The Arabids, Noctoideae. They're in India. There's been cases of outbreaks of kind of really chronic illness or sort of acute illnesses related to caterpillars feeding on fig trees from this genus. So it's likely that the frass is pretty horrible if you inhale it in large amounts. So you have to be quite careful throughout the study. But it was a good indication for potentially interesting results in terms of research. And I'm just going to go through the experimental design. So we basically had 3 species of fig, 2 species of moth. We took 10 leaves per tree. So we studied two trees per species. You can see a bit more detail on the next slide. And for some of these, we reared caterpillars, and we basically collected their frass at the 5th instar. So we collected caterpillar frass. And of course, these caterpillars were then able to rear them to adults. So we could collect the frass from these caterpillars and rear them to adults. We could take their wings, body, and other bits of the body to analyze. Some of the caterpillars we had to rear to stave as caterpillars. So they're from the same trees, but we can't directly link these individuals to those apart from the tree that they're feeding on, if that's clear. And what we did is we reared some caterpillars and we starved some, and we collected the frass from the 5th instar. This is after a long study of working out what if these caterpillars would Dyer's law of instar sizing, and we did this across 3 species of fig. So we had 6 trees for our preliminary instar project. For the main project, we had 6 trees from 3 species, and we took 10 leaves. We had 2 treatments for the caterpillars. This was either to let them develop to adulthood, or to take their or to keep their frass, and then to take the caterpillar. And in total, we had 476 samples. So this also includes various body parts that we took from the caterpillars. And I got this data on Monday. I got these data on Monday, so there's a lot we can still do. So it's a bit, it is recent research. There's some action shots. So we found it was much easier to rear these caterpillars on the tree than to take the leaves off. So we had a lot of trees covered in mesh bags, and we a bit more on the chemical analysis. This was done by a guy called Juha-Pekka Salmon. He's a professor of natural chemistry, leader of the natural chemistry research group in Finland. Here he's in action. He's constantly developing new methods to study plant chemicals, and one of the methods that we used in this study was kind of much newer than the previous studies, and it's metabolomic type approach, in that you quantify all sorts of small compounds. It's really hard to quantify big things like tannins, but we quantified 214 compounds from different groups of polyphenols, alkaloids, glycolates, and cyanogenic glycosides. About 43 of these were alkaloids, so that's more than we found in previous studies, but the methods are different. And this is what happens if you just do a CCA. So here you've got the open circles represent chemicals, so chemical composition, so species in a traditional sense. The closed circles represent the samples, so each sample taken, the sites. And then we've got the correlations between these variables across different types of samples. So the red are the adults, the green are the caterpillars, the blue are the caterpillars. Some of the caterpillars are parasitized, not intentionally, but kind of produce an interesting result. Then we've got frass and leaves. So this is chemical similarity across these different parts. And if we break that down to just the alkaloids, so obviously there's a lot of information there, and you get a really nice spread. Alkaloids, the spread's still pretty good. There are basically three different types of thing that happened. So you had compounds, you had different groups. You had compounds that were really like super, remember this is a log scale as well, super abundant in the adults, but not present at all in the leaves. So these hydrazine alkaloids are not different between tree species. Interesting, there's no effect of the tree species that the caterpillar was reared on. So they kind of converge actually. So despite the different history of their rearing, they produce the same compounds which aren't present in the leaves. So this to me suggests maybe an example of synthesis of a novel chemical. You have other cases where the adults try their hardest to get rid of these compounds, but they're present in the leaves and the frass. So these are probably compounds which are not beneficial. The adults don't want to sequester them, so they kind of pump them out in their frass. They try to get rid of them. And there are other compounds which distinguish really basically very different between groups of species of fig. So this benzyloquinone, which is an important alkaloid from the elevational transect study, is really very rich in sepsica, but isn't found in the others. So we've got all sorts of things going on. And remember, this data were collected very recently, so I haven't really had time to digest it all, to be honest. But frass and leaves are very chemically similar, and many compounds occur across tissue types. So a lot of these alkaloids are present in it everywhere. So there's probably a degree of tolerance. But caterpillar chemistry reflects host chemistry to some extent, but the adults tend to kind of fall in adults reared on different species. Also remember that these are different species of Asota as well, so there's a lot going on that needs to be picked apart, and these data are kind of under analysis at the moment. But I wanted to give you some insights as to how you can go from having these huge biodiversity studies to be really looking at the nuts and bolts, the details of how these interactions play out in nature. So Asota are probably capable of tolerating, detoxifying, sequestering and synthesizing alkaloids used in aposematism. The caterpillar and the moth chemistry reflect host chemistry, but there are different effects in the adults and the caterpillars. And large-scale forest studies can kind of throw up these really nice examples, and we've been very lucky to be involved with Binnison Research Centre, and the fact that they've got these resources and this incredible expertise. These guys are incredible field ecologists, often much better natural historians than many of us, and we've been able to do some of these follow up some of these lines of inquiry. So just to say really very quickly, and this is a bit of a plug I guess as well, I'm working on a proposal at the moment to take a similar approach in coffee, so wild coffee. This is a really nice figure, it's not mine. But there are 124 species of coffee, and these are what they kind of span across this wet dry gradient from West Central Africa to Madagascar. And the herbivory pressure probably also follows a similar gradient. So what we'd like to do is to try and study some of these wild species of coffee, their insect communities, and hopefully relate that to what's going on in pests on the cultivated species, so Arabica and Robusta. So that's I haven't really got time for much more. Just like to say thanks to everyone involved, especially the father-son combination of Bruce and Emil. He's looking a lot younger in this photo. That must be quite old. Yeah, I've got some references there. I'll leave those up, and I'm happy to take questions. I must admit I haven't been following the questions that have been coming up because I've been too busy trying to keep to time. But I'm
right,
sure I can work them out now.
Yeah. Thanks, Tim.
Okay.
I think people should ask their questions. Don't be shy. There's already a question by Raquel Vasconcelos. I seem to remember Raquel doesn't have a microphone from last week if it's the same person. Otherwise, you can ask the question directly. Raquel, are you around?
Okay, she said, could you please tell me the reference?
Yeah.
The map of diversity classes you presented in the introduction.
Mm-hmm.
So I have to just remind myself. Okay, so this map here is from, I guess this is the one, if not, the reference is down there in the corner. And I should reference that properly at the end actually, but I've kind of come to use it so much. It's a really, really great piece of work. Is that the reference?
I think Raquel can react if you want. You can react or expand on your question.
Yeah. Okay. That's it. Cool.
Okay.
Can you open your microphone?
I think she's okay. She said it's okay.
Okay.
So, while we wait for people to ask some more questions, I had a few questions myself. I think you touched upon it towards the end, in fact, but I had a question at the very start when you were doing this phylogenies and then looking at the influence of traits of different or chemical traits of plants on insect communities. I was wondering if it's possible to do some match with the insect side, or if there are some traits like that can be typed at the whole community level, perhaps for the insects. So I don't know, perhaps some of them would be detoxifying things, others would be whether, as you were showing, whether you find the same chemicals inside the insects, or I don't know. I'm not a specialist of that, but I'm just wondering.
Yeah, no, it's a good point.
So, done as perhaps a technique.
Yeah, so I mean, we wanted you can see that this is like a terrible thing. This is a figure from a slide from a presentation I gave in 2014, and these things were still to be done. This was done, never included, but this never happened. But one of the things we wanted to do was to take some traits from the adults, and so geometrids illustrate that point really well, actually. So in Europe, there are the autumn moth. Well, there's a load of quite polyphagous geometrids that are a big problem for apple producers. They like rosaceae a lot. They tend to go for quite polyphenol rich hosts. So a lot of the things they go for are rich in polyphenols, and there are some papers that suggest that actually the polyphenols that they take in are quite good for controlling things like pathogens and infections. So there's a lot more going on than this, and I think including some of the more of the information of a Lepidoptera, even things like body size would probably be quite important. You can see here there's a load of this group here. These are all pyraloid moths, and they're all feeding. This is basically ficus, and in geometrids there's nothing really eating ficus, but in pyraloids there's a lot of stuff eating ficus, and these moths are quite distinct in their natural history and their traits. So I think even within these two groups, there's a lot of mileage for studying basic life history traits.
Yeah, there's this, but then I was thinking, just technically speaking, is it the same thing to do this chemical analysis on plants and or insects, or is it a different?
Yeah, so for the last study, we basically, so if you can freeze dry it and you can crush it up, then you can analyze it. Basically, I say that on behalf of you, Apaka, but I think that's pretty much the cut and thrust of it. Yeah, so this would be fascinating, really, to do a kind of whole family level chemical ecology of the insects of the hosts and their insects. That would be amazing. Actually, yeah, that'd be lovely to see. Because there's clearly a lot of interesting chemical ecology going on at the kind of species level that it's really hard to fit into these wider studies. So this took a long time, for example, but I'd like to see that.
Other questions? Are there some explanation for the covariance of chemicals with elevation? Is it just because you think it's some kind of, or perhaps you said it, is there some kind of just random association of some trees with elevation, or is there some things that make some chemicals more difficult to do at different temperatures or things like this?
Well, we think that some of these chemicals are probably, so there's this debate, carbon nitrogen debate in plant chemistry, that nitrogen is needed to make some of these compounds. Of course it is, but whether these basic resources and elements change with elevation is one possibility. But I think that's probably not a limiting factor. I think if I was going to bet, I think a lot of this stuff is abiotic. So, for example, the flavonoids are probably, I would say, largely for UV protection. So it's quite a steep gradient. It gets up to about 2,700 meters is the highest elevation we sample at. And you've got quite high levels of UV up there. So I think a lot of these things are in response to the abiotic environment. There are biotic drivers that we just haven't measured. So we do, in the paper, we do count marsupials, and there's a trend which roughly correlates with the non-flavonols, but it's kind of just a correlation. But I think fungi are probably also pretty important. So a lot of the fungi, the fungi that grow on the side of these trees probably drive the alkaloid content. So I think there's a few biotic factors and abiotic factors that we haven't measured. I don't know if these things are just simply harder or easier to make. There's probably a trade-off with growth and regrowth and tolerance. So maybe these things are more costly. Some of the alkaloids are costly to make, but it's more if you're high in elevation, because if you lose your leaves, it's much harder to regrow that material. So you defend it as best you can. But I don't know if any one of these traits is specifically more costly than others, or kind of resource dependent.
Yeah, but it's interesting if some, I mean, you could say perhaps some of these chemicals have some abiotic drivers selecting for them, and then they have a side effect on.
Yeah, I think that might be the case with some of these, these proanthocyanins. So these are the similar class to the that you get in leaf senescence and these colorations, and they basically the pH of the insect gut is alkaloids, and some of these tannins work really well. Sorry, these oxidative chemicals, tannins especially, work really well in that kind of particular chemical environment. These other groups don't really do much they don't bind proteins in the way that they're meant to, if they would be for insect defense. So we think they're probably more of a defense against, generally probably a defense against some of these mammalian herbivores. And actually there's a really recent study where they took some of the diets of these insects and these mammals, and they discovered that yes, there were lots of compounds in the leaves, but that also, yeah, this was mostly to do with kind of carbon and nitrogen and phosphorus and stuff. But it would be really cool to try and link the protein precipitation capacity, which is meant to be a direct response to mammalian predators, to some of these larger mammals. So I think, yeah, the insects probably have a role, but there's a lot more going on, fungi especially. But
so, do we have some question by some specialists we have some chemical ecology people in the audience, or I guess community ecology? Finn, any question?
Yes, maybe a small question on the geographic variation. What is your expectation? You have you had some very neat data on Ficus septica, and that is a species that covers a huge range. So anything that you expect on the variation of the chemistry there?
Yeah, that's a really good point. You're totally right. So Ficus septica is it's got a huge range. Some of these others, so Hispanoides doesn't. Pachyrhachis is an endemic to New Guinea. Septica definitely isn't, and that would be might be quite a neat candidate for testing if this truly is an anti-herbivore defense, for testing if there's a gradient in herbivory, for example. So septica gets all the way up to Philippines or further.
Oh, it goes up to Taiwan and to.
Yeah. It's in China as well, isn't it? In Sichuan Valley.
Okinawa.
Okay, so if there is a gradient in herbivory, for example, that would be a really nice system to study on that along that gradient. My kind of gut reaction would be that there probably is a difference, and that should correlate to historical levels of herbivory. So I guess the classic interpretation is that this is a wet forest, wet humid evergreen forest with high rates of herbivory, and that it pays off to invest quite heavily in plant defense. But maybe the same defenses aren't needed as you get further north in the range, and rates of herbivory decrease as you get to as you go slowly up towards the subtropics. It's also in Australia as well, so you could probably study the species in some of these drier habitats as well. But that would be my feeling, that the chemistry probably does vary. I think these fundamental molecules are still produced, but perhaps in different concentrations. But having said that, this interaction, so Asota is in Sichuan Banner, there's quite a lot of Asota species, and Asota gets all the way up to northern India on figs. So if it's a really long co-evolved, long existing interaction, then perhaps septica is similar in all places.
All right, so there's a question by Martin Hossert. Martin, do you want to ask a question yourself to make it more interactive, perhaps? It's about, so Simon, you can see the question. It's about.
Okay, so I'm just going to find my way back into the Q&A.
Just if Martin wants to react, she can turn on the microphone.
Okay.
Known about the alkaloid detox.
Yeah, so what is known about alkaloid detoxification and sequestration in Asota?
I think Martin's microphone is on now.
Okay.
Okay.
Yeah. But you have the question right now, so you can answer.
Just if you want to react or something.
Yeah.
Yeah. So actually not very much. So there's been a series of studies on their host plants, but not because they're the host plants of Asota, but because they've got lots of alkaloids by groups for a while. So we know that they're alkaloidal. There was a study. I have to dig it up. We reference it in this paper here, this Wolf paper on the elevational gradients. But there's a reference I think it's in +1, where they show that there's an outbreak of really kind of acute respiratory problems in a part of India when these moths are feeding in high levels on local fig trees. So there's a medical condition that kind of responds probably to the chemicals in the frass of Asota. So that's just a kind of observation, really. That's what got us thinking that maybe there's something going on. I think there's actually very little known about the mechanisms, about how Asota does it, about what exactly how these compounds are changed as they go from leaf to adult, or how novel compounds are modified from basal precursors. Basically nothing. So the nice thing about this data set is that these kinds of data are there. So we have the chemical structures, the formulas of all the compounds in the leaves, and what we're hoping to do is to trace some of these chemicals, these synthetic pathways from precursor to product. by taking this kind of comparative approach. Obviously, the ideal way to do it is to label each compound with an isotope, but this is quite a focused way of doing it, because you can't do a whole community per se of chemicals. So the short answer is not much, but hopefully soon we'll be able to have a bit more detail about the exact mechanisms.
Okay, thank
So,
you.
sorry.
Wait for that.
Yeah, it's a really new data set, but it's
Yeah.
for me it's one of the most interesting, because it was an opportunity to really go from something that we'd observed in the field and these big patterns to kind of pin down the natural history and the chemical ecology of an individual organism. But yeah, no, I'm really excited by these data. And of course, there's all these other things that aren't alkaloids. So there's, in the first plot, you can see that the separation is actually really, really strong. So there's huge separation between species and tissue type. And then when you go down to just alkaloids, some of that separation kind of is less distinct. So some of these things will be amino acids, which obviously are more present in caterpillars, but some of these other things are polyphenols and glycosides, stuff that the caterpillars may well try to sequester or may well kind of try to excrete as part of their metabolism. But yeah, I really kind of excited about these new chemical methods. But I think as long as they're used with kind of real care and expertise, and you kind of know exactly what's going on, I think they can be incredibly powerful.
Okay, so I think we'll end here because time is running. People might want to go to lunch. So I want to thank Simon again for this nice talk, and then tell everyone, see you next week. We have a few more talks until the end of June. So have a nice weekend. Bye-bye.
Okay, thanks very much. Thank you, everyone. Thanks for listening.
Bye-bye.
Bye.
Bye.
Okay, thanks, guys. Thanks for that.
Thanks to you.
Hey, great talk.
Thank you. Yeah, sorry, I messed up one of the slides. It's sort of those things that must have been, I just missed it. And then if you have one mistake in a slide, it throws you off